Extracellular vesicles are small lipid-bound vesicles that shuttle proteins, lipids, and nucleic acids from one cell to another. From studies in diabetic mouse models, we know that extracellular vesicles can affect gene expression in macrophages and are mediators of inflammation, but their role in humans is not clear. Can diabetic extracellular vesicles affect gene expression of inflammatory response, oxidative stress, and apoptosis in human monocytes? Remember, these pathways are characteristically altered in diabetes.
Conducting a microarray analysis, researchers found that diabetic EVs did have an effect on the previously mentioned pathways of monocytes. To verify these results, a qPCR test was conducted to measure the expression of specific genes involved in these pathways. Firstly, monocyte cells were incubated for 6 hours in medias containing either diabetic EVs, euglycemic EVs, or no EVs. Following that, the mixture was centrifuged to separate the media and monocyte cells. Total RNA was then isolated and checked for quality and quantity. cDNA was obtained using reverse transcriptase and random primers. Then, a qPCR test was conducted on the cDNA using gene-specific primers and SYBR green as that reporter. The expression of target genes was normalized to household genes HBRT and UBC. All the necessary protocols were provided and referenced, making the methods easy to follow and reproducible. The results of the qPCR test can be seen in Figure C. It is shown that diabetic EV condition resulted in higher CXCL5 gene expression in monocytes when compared to the euglycemic condition. Also, genes DDIT3 and CRAG that are involved in the apoptosis pathways and genes GSTP1 and SAW2, which are all involved in the oxidative stress pathways, experienced lower expression in the diabetic condition. A weakness in the results was that the method of measurement of relative expression, seen here in the y-axis of the figure, was not mentioned in the study. This led to some confusion amongst readers about how these values were obtained. Aside from this, the figure and the results sections did a good job in expressing the significant outcomes.
In the discussion, the authors suggested that the changes in inflammation, oxidative stress, and apoptosis pathways stem from the preferential internalization of diabetic EVs in monocytes. So the upregulation of the gene involved in cytokine secretion could explain the increased inflammation seen in diabetes. Also, the decrease in apoptotic gene could lead to the impaired monocyte apoptosis, interfering with monocyte differentiation in diabetic patients. At the same time, downregulation of genes encoding for antioxidants can decrease diabetic individuals' ability to combat oxidative stress. Overall, qPCR helped the authors effectively demonstrate the role of EVs on the intracellular pathways of monocytes in human diabetes.